At the very earliest stages of development, all cells are equally capable of giving rise to an entire organism. But after only a few rounds of cell division, the cells begin to lose this totipotency and to differentiate, sorting into groups that will either contribute to the placenta or to the embryo itself. In this 8-cell embryo, called a morula for its berry-like appearance, all cells express a pair of genes, CDX2 and OCT3-4. But as the cells of the morula go through their next round of division, CDX2 is downregulated in the innermost cells, while OCT3-4 remains expressed throughout. The segregation of these genes is maintained as the cells continue to divide, until after about 10 rounds of division, the embryo has become a hollow ball, known as a blastocyst. The outer layer of the blastocyst is made up entirely of cells that express CDX2. This trophectodermal layer will go on to become placenta. On the interior, OCT3-4 marks a small cluster of cells with phenomenal ability to differentiate. This inner cell mass is made up of cells with the developmental potential to give rise to all of the body's many cell lineages, an ability known as pluripotency. And indeed, every one of the cells in your own body can trace its ancestry back to these few dozen pluripotent stem cells. The vast majority of the body's trillions of cells are differentiated, meaning they have taken on specialized properties and functions, and have lost the ability to generate cells of other types. Skin, muscle, blood, bone, and the nervous system are all made up of populations of differentiated cells. There is another type of cell, however, that remains less differentiated and retains its ability to give rise to other cell types. These cells are known as stem cells. Stem cells exhibit two characteristics not found in more differentiated cells. By definition, stem cells are able to divide and make copies of themselves indefinitely, a capacity known as self-renewal. This endless power of division is not limited to creating more stem cells, however. They can also give rise to a wide range of mature cell types. These unique characteristics make stem cells indispensable, both for replenishing the body's cells as they age and are lost, and for constructing the body itself from a tiny number of cells during development. Even after the embryo has grown into an adult, many of the body's cells continually die off and need to be replaced. For example, billions of red blood cells need to be replaced each day. This is the work of stem cells known as hematopoietic stem cells, which are located in the bone marrow. Residing in a complex cellular environment, the hematopoietic stem cell is able to generate precursors to every type of blood cell. And indeed, these stem cells have been used by physicians for decades to replenish patients' blood supplies following radiation treatment for cancer. The stepwise differentiation of a stem cell's progeny depends on combinations of genetic and environmental factors that gradually steer these cells into following a specific lineage. This process involves different sets of stimulating factors operating in the cell's environment, at its surface membrane, and within its nucleus. Each step down the path draws it closer to becoming a functional red blood cell and entering the bloodstream. The lining of the small intestine turns over even faster than the blood. At the microscopic level, this epithelial surface consists of billions of projections called villi, with stem cell-containing areas known as crypts near their bases. The intestinal crypt contains slowly dividing stem cells that maintain their own numbers while also producing rapidly dividing progenitors. These transit-amplifying cells churn out all of the differentiated cell types needed to migrate upwards and replace older cells as they are shed from the tips of the villi. This constant self-renewal by stem cells is a feature of many of the body's systems, including skin, hair, and bone. Not all of the body's organs regenerate as readily as the blood and the lining of the gut, and the multipotent stem cells that have been identified in the adult body are both limited in their ability to generate other lineages and difficult to maintain in culture. In contrast, the pluripotent cells of the inner cell mass can be harvested and grown indefinitely in vitro, and can give rise to every type of cell in the body. These pluripotent cells in culture are known as embryonic stem, or ES cells. By culturing these cells under controlled conditions, it is possible to steer their differentiation into specific cell types. For example, ES cells cultured on a bed of stromal feeder cells and treated with inductive factors such as BMP4 and sonic hedgehog tend to differentiate into neural lineages. By testing various ES cell culture conditions, scientists are gaining new insights into the biology of the differentiation processes that build and sustain and may one day be used to heal our bodies. Stem cell researchers are currently exploring ways to differentiate ES cells into lineages that are of great medical and biological interest. The nervous system, the pancreas, and the heart all regenerate too poorly to restore themselves after serious injury or degenerative disease. Scientists are now seeking ways of using the knowledge they gain every day from the study of embryonic and somatic stem cells to help save patients' lives and to improve our understanding of their seemingly limitless potential.
Now, imagine this is a stem cell.
Stem cells are unique because they have the ability to generate new cells of almost any kind. They all start off as unspecialized cells, but
given the right chemical and genetic signals, the stem cells can divide to form slightly more specialized cells of different size, shape, and function. And after a few more cycles of division, these can give rise to highly specialized cells, like heart muscle cells, for example, that help your heart pump the blood around your body. Given a different set of signals, this same unspecialized stem cell can go down an alternative pathway and give rise to a different type of specialized cell, like a neuron that transmits electrical signals in the brain.
compared with stem cells from embryos, adult stem cells give rise to a smaller number of cell types, usually those of the organ or tissue in which they're found.
Now, researchers have found adult stem cells in more tissues than previously thought: bone marrow, skin, brain, liver, eyes, and this has led to research into using the patient's own adult stem cells to repair damaged organs.
Just bring your arms back your head.
And that's exactly the focus of the trial Michael Taylor is taking part in. Led by Professor Anthony Mather at the London Chest Hospital, the trial is vitally important because heart disease is still on the rise.
Cardiovascular disease is the #1 killer in the UK, with nearly 3 million people suffering with heart conditions in the UK, and about 800,000 people suffering with this condition of heart failure. He's got a condition which makes the heart very weak and baggy, and it doesn't work very well as a pump. And what we're trying to do is see whether his own stem cells can actually repair that heart and make it pump effectively again.
So if you're looking to repair heart tissue, do you need to use heart stem cells?
Ideally, yes. The problem is that there are very few of those cells in the heart, and we've only recently discovered that they're there. And the few heart stem cells that are present clearly aren't capable of repairing it. And that's really where we step in with our trials. By using bone marrow as a source of stem cells, we try and enhance the stem cells in the heart and their ability to repair the damage that's been caused.
This trial tackles heart disease on two fronts. Michael's bone marrow has already been stimulated to release enormous numbers of stem cells into his blood. And now, more are to be harvested directly from the bone marrow in his hip.
You okay?
Yeah, fine, thank you. Yeah, it wasn't that bad at all.
Good.
As this is still a trial, only some patients will have these stem cells re-injected. Although Michael will get an injection directly into his heart, neither he nor Professor Mather know if it will contain his stem cells or a placebo.
We're about to put either stem cells or placebo into his coronary arteries. We won't know which until the end of the study when we've treated all of our patients.
This uncertainty helps the researchers to rule out the placebo effect from any positive results they achieve. Professor Mather feeds a tube through Michael's blood vessels right to the site of the disease in his heart. Incredibly, Michael remains fully awake throughout the entire procedure.
All right there, sir?
Yes, fine, thank you.
Good. Can't feel anything?
No, I can't feel anything.
So that's the first artery done, now the second one, and then one more, and then we're all finished.
In less than a day spent in hospital, this patient's eyesight will be restored. Stem cells from the patient's own eye have been harvested on this simple contact lens. This will be placed on the eye to overcome the damage caused by blinding corneal disease. Within weeks, sight will dramatically improve. It's a world-first technique pioneered by researchers at the University of New South Wales and colleagues at Sydney's Prince of Wales Hospital. The novel procedure, which is simple and inexpensive, offers hope to people with a range of blinding conditions that involve corneal damage from infections, burns, and chemotherapy.
It's a simple technique. It requires a small biopsy from a patient. It requires 5 mils of sera. The cells are cultured over a 10-day period on a contact lens, so it requires no foreign human material or animal products. During the culture procedure, the tissue is firmly adherent to the contact lens surface. The pink solution is like fertilizer for the cells. The cells seem to transfer from the contact lens surface onto the patient's ocular surface.
Once the stem cells have multiplied, the lens is dispatched to surgeon Dr. Stephanie Watson of Sydney's Prince of Wales Hospital.
The procedure is relatively non-invasive. For the patient, they merely come into the hospital for a couple of hours and then they're able to go home afterwards. At the beginning of the procedure, we remove all the damaged cells from the front of the eye. Following this, we take the contact lens with the cells that's been cultured in the lab and simply place it on the front of the eye. Afterwards, the patient uses eye drops and then returns the next day and we examine the contact lens to make sure it's in place.
The researchers are hoping the procedure can be adapted for use in other parts of the eye and even in other organs.
We're very excited about this technique because we think it might be applicable to other major organs of the human body, such as the skin, because after all, the skin behaves in a very similar manner to the cornea.
It's being called a medical breakthrough, and it offers great promise to patients suffering from serious illnesses. Only a handful of windpipe or trachea transplants have ever been performed, but this is the first to use a lab-grown airway. Claudia Castillo suffered from tuberculosis for years. Then in March, she suffered a serious lung collapse. Initially, doctors thought the only solution was a risky surgery to remove the entire left lung. But a team of surgeons proposed a windpipe transplant with stem cells instead. Once doctors had a donor windpipe, it was stripped of all its cells. Then doctors took a sample of Castillo's bone marrow from her hip. Stem cells from the bone marrow were then used to create millions of cartilage and tissue cells to cover and line the windpipe. Doctors were then able to perform the transplant.
The state of Claudia is excellent. She is enjoying her life. She has returned to be mother. As long as she stays in this health, I will be the most happiest doctor in the world.
Since the surgery in June, Castillo hasn't shown any signs of rejection, and she's not taking any immune-suppressing drugs, which can cause serious side effects. She's now able to take care of her 2 children and can even walk reasonable distances without becoming out of breath. And doctors are hopeful that one day the technique can be used for other organs, like the bowel, bladder, or reproductive tract. Sandy Kozel, The Associated Press.
And this morning's Health Watch, healing hearts. This morning, researchers are reporting what could be a major breakthrough. Cardiac stem cells can repair damage from a heart attack.
Correspondent Bill Whitaker reports on one man whose life has been changed by the new promising treatment.
So Ken, how are you feeling?
I feel fantastic.
When we first met Ken Millis 2 years ago, he had suffered a serious heart attack. At age 39, 30% of his heart was damaged. He faced a shortened life with shortness of breath and restricted exertion.
If someone's had a heart attack and they have seen part of their heart turn into scar, that is irreversible. It's there for life.
So Dr. Eduardo Marban, director of the Cedars-Sinai Heart Institute in Los Angeles, convinced Ken Millis to be patient number 1 in a risky, first-of-a-kind clinical trial. Dr. Marban used a catheter with tiny pincers to snip bits of healthy tissue from Millis's heart. In a laboratory, he coaxed the tissue into manufacturing millions of stem cells, which were then deposited in the damaged area of the heart. Millis was one of 25 trial subjects, and the results are now in.
The subjects who received cells had a dramatically better response than the control subjects.
All the stem cell recipients had stronger heartbeats, and their heart attack scars were reduced, on average by almost 50%. Damaged muscle was replaced by new, healthy heart tissue. Ken Millis had better than 50% improvement.
It's the very first time, in my experience, that we've actually exceeded our expectations when we do a human trial.
Doctors say Ken Millis's heart looks almost normal again.
It's like someone gave me a magic pill. I felt better all over, suddenly. You know, it's science fiction, basically, to me. That's what it feels like.
These are just the first results of a unique study. Results cardiologists find promising. Bill Whitaker, CBS News, Los Angeles.
And here with us now, Dr. Holly Phillips. Always nice to see you.
Great.
So give us an idea, because it sounds promising, of course. How could this treatment actually revolutionize the treatment for heart failure?
Well, you know, Erica, it can make a dramatic difference because we've never before thought of heart disease as a curable or as a reversible condition. The treatment has always just focused on minimizing symptoms and making living with heart disease better. This completely changes the landscape of how we think about the illness. The other thing is, this is the first time cells from the heart have been used to treat the heart. Before, we've attempted to treat the heart with stem cells from bone marrow, but this is the first time they actually come from the organ, and so that gives us hope for possible other organs. Maybe we can use cells from the kidney to treat the kidney, or cells from the liver to treat the liver, and so forth.
So we're looking at about 6 million people in the country that have heart failure. What exactly puts you at risk for this?
By far, the most common risk factors are coronary artery disease, you know, really heart disease, and heart attack. That causes the vast majority of heart failure symptoms. Also, high blood pressure, diabetes, and even sleep apnea can contribute. So there are a number of factors that can cause heart failure. But once you have it, it is a very serious illness. 1 in 5 people who are diagnosed with heart failure die within a year of diagnosis.
Really quickly, I have about 15 seconds. If you have been diagnosed with that, how can you keep the condition from worsening?
There are many ways, but probably the single most important thing, if you only do one thing, it's to quit smoking. Smoking raises your blood pressure and makes your heart beat faster. Both of those things stress your heart and can make heart failure symptoms much worse. So that's the one thing you have to do.
All right. Dr. Holly Phillips, thank you. Good to see you as always.
In the 2005 sci-fi thriller The Island, people have found a way to live forever. They grow clones and harvest their organs. But real science may be on the verge of a less diabolical solution. This, for example, is no special effect. It's a lab-grown lung, no clone attached.
I absolutely see a day where you'll walk into a manufacturing facility somewhere and there will be jars of kidneys and jars of livers and jars of lungs, whatever it is you need.
Just as in The Island, your body would accept the new organ because it would be yours, grown from your cells.
And there would be no more waiting lists for organs. There would be no more rejection. We would enter a new era where we could build you an identical, ideal replacement.
But how do you make an organ without a body to build it in? We've been growing cells in the lab for decades, but they just sit around in flat layers or clumps. So how would you coax them to form a three-dimensional organ like a heart, with chambers, valves, and blood vessels?
Maybe it's the same way you go from this to this. See, an organ is not unlike a building. It's a collection of parts that has to come together and work together. Think of a cinder block as a cell. The problem is, a block or a cell alone is not enough.
To construct a building, you need to begin with an internal framework or scaffold to define the parts and hold them together. 30 years ago, transplant surgeon Jay Vacanti and chemical engineer Robert Langer realized that to build an organ, cells also need a framework, a scaffold to guide their growth. The challenge was to engineer scaffold materials living tissue could grow on.
So this is a material that we call biorubber.
Biorubber, and you use the prefix bio because whatever is the material it will take to flesh or living cells.
That's right.
So why does the cell even care?
Because a lot of things could be toxic to a cell, or the cell wouldn't like their surface and wouldn't be able to grow on it.
Picky cells.
Cells are picky, and some are more picky than others.
But sculpting a scaffold out of the right material is only a start. To turn one into a living body part, an ear, for example, it must then be seeded with cells. A few weeks in an incubator allows those cells to multiply, covering the scaffold. Then comes a rather strange test.
This is really creepy. I mean, mice are creepy enough, and this one has no hair and a human ear growing on its back.
Yes.
He doesn't seem to mind that he has an ear growing on his back.
No, he knows he's here for a bigger purpose. But this is a very, very important step in the science, because on the back of this animal, we're actually incubating and growing perfect cartilage in the shape of a human ear. And it's completely connected to the blood vessels, so that it's just like a native ear in a normal circumstance.
In the head of a person.
That's correct.
So when this finally gets implanted in a human, you don't expect rejection, as is so common with new body parts.
Exactly, because we're going to start with the patient's own cells. It'll make his own tissue, and therefore the body will accept it.
Within a year, Vacanti and Langer expect to be implanting their ears directly on the heads of soldiers wounded in Iraq and Afghanistan. But these will not be the first recipients of lab-grown body parts. Already, patients of other doctors have received blood vessels, skin, muscles, even bladders built the same way.
I think with enough research, most parts of the body will be replaceable. And I haven't come across very many body parts where somebody somewhere isn't working on trying to replace them.
Which is certainly encouraging news for people who need more complex body parts, like 20-year-old Stacy.
I was in the hospital, and that's when they came in and told me that I may need a new liver.
But will she get one? Every day, nearly 20 Americans die waiting for donor organs.
So this problem is an extraordinary problem. There are too few organs for the well over 100,000 Americans waiting.
But if we're ever to make the complex organs most needed to save lives, like livers and hearts, the scaffold builders will have to overcome an obstacle, namely plumbing. In a building, it's pretty straightforward. Pipes carry fluid where it's needed, just like blood vessels in the body, except that in a major organ, like the heart,
You need a blood vessel per cell because the heart works all day, every day. And I don't know if you've ever seen blood vessels, really, but they look like a tree. And the challenge is not to build that big limb, but to build those little tiny branches that come off.
But building these intricate branches might be unnecessary if we take advantage of a remarkable fact. Organs are not just made of cells.
So if you wash the cells away, what's left? Well, what's left are these proteins on which the cells sit, and they form the framework of the organ, the scaffold.
These natural scaffolds hold an organ's shape down to the smallest detail, including every blood vessel. So could they be used to build a complex organ like a heart? 6 years ago, no one could say, because no one had ever stripped a heart of its cells, leaving the scaffold intact. But Taylor's colleague, Harold Ott, thought he could find a way. He would use the blood vessels in a rat's heart to deliver a chemical that would dissolve its cells and nothing else. But which chemical?
So the process of finding the right chemical was literally a try and error process, starting from A to Z on the chemical shelf.
First, Ott tried enzymes, but they dissolved both the cells and the scaffold. Other chemicals caused the hearts to swell up. Finally, he tried a soap commonly found in shampoos.
We saw the heart become translucent, and it was obvious to us all that something had happened that hadn't happened in the months before.
What we had is this thing that looked like a heart, but it looked like a ghost heart, if you will.
Injections of dye showed the scaffold to be undamaged, down to the smallest blood vessels. And we now know that this technique works with many organs, including human-sized ones.
This is essentially the scaffold of a heart. Who knew a heart had a full skeleton? But it essentially has no cells, dead or alive. It's beautiful. You can see the blood vessels here, the chambers of the heart. You can see the valves.
But could a bare scaffold once again become the framework of a living heart? Taylor soon discovered it was more than a matter of injecting cells.
Just putting cells on a scaffold isn't enough. It's putting cells on a scaffold and giving them an electrical signal and giving them a mechanical blood pressure and then giving them oxygen. It's not just a heart in a jar, it's a heart in an artificial body. So it's simple in many ways and it's unbelievably complicated.
After 8 days, the first lab-grown heart beat on its own.
It really makes you go, what is life? The first time you see something beat that was dead, it's one of those yes moments in life.
Since then, Ott has joined Massachusetts General Hospital and used the same method to build a pair of lungs. After coming back to life, one lung was successfully implanted in a rat.
So if you can make a working, living lung, then it seems to me you can
build literally any organ.
Any organ.
This novel approach has already made a difference in the real world. In Barcelona, Spain, this woman, Claudia Castillo, might be dead without it. 2 years ago, tuberculosis devastated her windpipe, making it difficult for her to breathe. But surgeon Paolo Macchiarini saw a solution. would give Claudia a new windpipe, which her body would never reject, because it would be made of her own cells, grown on a natural scaffold. And so, in June of 2008, Macchiarini and an international team of specialists removed a windpipe from a human cadaver, washed it clean, and reseeded it with living cells from Claudia's body. 4 days later, the new windpipe was transplanted into Claudia.
If you transplant an organ without tissue engineering, you need immunosuppression, you need close watching, and this was absolutely not the case for Claudia. She never had any sign of rejection. Indeed, 4 days after surgery, she was home.
More than a year later, Claudia is living a normal life, free of the fear that she will reject her new body part.
I feel like the transplant is not from the body of another person. It's mine.
That sense of ownership might soon be crucial to organ recipients, because their scaffolds might not come from a person at all.
This is a pig kidney sliced in half, and it's the same size, same complexity as a human kidney. We could cover this with human cells and, in theory, build you a kidney.
Human organs built on natural or artificial scaffolds? Made from a patient's own cells to avoid rejection? Available in unlimited supply? Most researchers believe it will be a reality within decades. And Taylor is even more optimistic.
Kidney, liver, lung. We're not decades away from building something complicated. We're more like years away.
This is the CBS Evening News with Katie Couric.
The casualty count from the wars in Iraq and Afghanistan continues to climb. By the most recent estimate, more than 30,000 American servicemen and women have been wounded. But now, an amazing development could change how doctors treat even the most serious wounds. As Wyatt Andrews reports in the second part of our series, Growing Miracles.
Good afternoon, Hobbytown USA, Lee speaking.
You might become a believer in the power of magic dust.
And I powdered it on until it was covered.
When you see how a special powder regrew the tip of Lee Spivak's finger.
I put it through the back of the airplane.
The half-inch he sliced off in the propeller of a hobby shop airplane.
Your finger grew back flesh, vessels, and nail.
4 weeks.
In 4 weeks.
Right.
Is this essentially what regrew Mr. Spivak's finger?
Yes, it is. We took this, turned it into a powdered form.
This powder is a medical product called extracellular matrix. Made from pig bladders, it's a mix of protein and connective tissue surgeons often use to repair tendons. But it's the matrix's unusual power to regenerate tissue that's helped launch a new field called regenerative medicine.
It tells the body, start that process of tissue regrowth.
At the University of Pittsburgh's Center for Regenerative Medicine, Dr. Stephen Badalik believes the matrix somehow mobilizes cells, some of them adult stem cells, whose job it is to maintain and repair injured tissue.
It will change the body from thinking that it's responding to inflammation and injury to thinking that it needs to regrow normal tissue.
If this helps Mr. Spivak's finger regrow, could you grow a whole limb?
In theory.
That theory that it might be possible to grow an entire limb is about to be tested by the United States military. The Army, working in conjunction with the University of Pittsburgh, is about to use that matrix on the amputated fingers of soldiers home from the war.
Can we make skin? Can we make bone?
Dr. Stephen Wolf at the Army Institute of Surgical Research says the military has invested millions of dollars in regeneration, hoping to regrow limbs, lost muscle, even burned skin.
It's hard to ignore that this guy is missing half his skin. This guy is missing his leg. Is there any way we can make that go back? Some of that technology exists.
Several different technologies for harnessing regeneration are now in clinical trials around the world. This machine, being tested in Germany, sprays a burned patient's own cells onto a burn, signaling the skin to regrow.
You could fashion this into a tube like this.
Dr. Badalik is about to implant matrix material shaped like an esophagus into patients with throat cancer.
So we fully expect that this material will cause the body to reform normal esophageal tissue.
Some of the most advanced tests involve the heart. This patch of material is being put on like a Band-Aid to regenerate tissue damaged by a heart attack.
Injecting one.
And here, patient Mary Beth Babbo is getting her own adult stem cells injected into her heart in hopes of growing new arteries. Her surgeon is Dr. Jun Lee.
It's actually, if you will, what we would consider the holy grail of our field for coronary artery disease.
The holy grail, because if stem cells can regrow arteries, there's less need for surgery.
If people don't have to go through that, this would be the way to go if it works.
This is only 2.5 years old.
Lee Spivak jokes he's got a 69-year-old body and a 2-year-old fingertip. But his fingertip has researchers imagining a time when regrown limbs replace prosthetics, when regrown tissues replace surgery, when the body does its healing with cells from within. Wyatt Andrews, CBS News, Pittsburgh.